Welcome to the second session of Human Health and Disease. In this session, we will mainly discuss immunity, different characters of immunity, and also the components of human immune system. First of all, what is immunity? Immunity is the overall ability of the host to fight the disease causing organisms conferred by the immune system. Immunity can be divided into two types. There is innate immunity and acquired immunity. Innate immunity is a type of immunity which is non-specific. Non-specific means it is not specifically identifying a microbe, and it is attained right at the time of birth itself. The second type of immunity is known as acquired immunity. Acquired immunity, on the other hand, it is specific immunity, and it is we can say it is pathogen specific. It is also characterized by memory. Innate immunity has got certain barriers. The barriers of innate immunity are: first, physical barrier, physiological barrier, cellular barrier, and cytokine barrier. So, what do you mean by these barriers? Only by crossing these barriers, pathogens are able to infect a person and they can cause a disease. So, all these four barriers that we have shown here, they are obtained right in the beginning of life or at the time of birth itself. So, we can say that these barriers are non-specific. Now, going to features of these barriers. Physical barriers. Skin on our body is a main barrier. Mucus coating of respiratory, gastrointestinal and urogenital tract, that is also a barrier. So, only by crossing the skin which is covering our body, a pathogen can enter, and also when it is trying to enter through a natural opening, it will be going through different tracts. So, all these tracts are lined by a mucus covering. So, skin and mucus will act as a physical barrier. The second one is physiological barrier. By physiological barrier, we mean the acidity in the stomach, saliva in mouth, tears, and also cerumen which is the ear wax. It is also a physiological barrier. Pyrogens are agents which are causing fever. They also come under physiological barriers. In our body secretions, as you know, there is lysozyme. Lysozyme is present not only in saliva but also in sweat, tear. So, this lysozyme will be antibacterial. It will prevent any attack of bacterial cell. It is able to degrade the bacterial cell wall and thereby acting as a physiological barrier for our body. Now, coming to cellular barriers. Cellular barriers mean cells as such acting as barriers. For example, polymorpho-nuclear leukocytes, also known as PMNL, they are actually modified forms of neutrophils. They are actually neutrophils. Monocytes, natural killer cells. Natural killer cells, they are a type of lymphocytes, and also macrophages. Macrophages are formed from monocytes. So, all these cell types which are involved in maintaining immunity, they are called cellular barriers. Now, the fourth one is known as cytokine barrier. By cytokine barrier, we mean substances that are produced by a cell and will be helping for maintenance of immunity. Interferon is such a substance. Interferon is a glycoprotein which is produced by host cell whenever there is attack of viruses. And these interferons are further preventing attack on unattacked cells. So, interferons that are produced by virus attacked cells or host cells come under cytokine barriers. So, these are the barriers. All these barriers belong to our innate immunity. Our acquired immunity is having two types of responses. What are the two types of responses? First one is primary immune response and the second one is secondary immune response. Primary immune response happens when an antigen enters into a body for the first time, and primary immune response, because it is due to the first exposure of antigen, our immune system is not prepared, so it is comparatively of low intensity. As you can see in this picture, you can see it is of low intensity, the first exposure of antigen. You can see in the plasma the antibody concentration is comparatively low. What about the secondary response? Secondary immune response will happen when the same antigen encounters for the second time. When there is a second encounter of the same antigen, our immune system is already prepared. I mean, already there will be preparation of memory cells. So, these memory cells will identify the antigen quickly and they are able to produce a heightened immune response or anamnestic immune response. So, that is shown here, and you can see the plasma antibody concentration is comparatively high. So, primary immune response is of low intensity and secondary immune response is of high intensity. There are two types of cells involved in maintenance of acquired immunity. There is T cell and B cell. T cell is involved in maintenance of cell mediated immunity and it is produced from bone marrow, but it will be matured in thymus. But what about B cells? B cells, they are formed from bone marrow and also they are getting matured in bone marrow itself. B cells, they are the cells that are involved in antibody mediated immunity. So, T cells, they are involved in cell mediated immunity, whereas B cells, they are involved in antibody mediated immunity. Here in this picture you can see how the humoral immunity or antibody mediated immunity works and also how the cellular immunity works. You can see it over here. The T cells that are activated will produce effector T cells known as helper T cells. So, here you can see the helper T cells are activating the B cell, and the activated B cell will produce the memory cell as well as the plasma cell. And here plasma cells are the cells that are freely wandering in the body fluid and they are involved in the production of antibody. And these antibodies, they will bind with antigen. They will produce antibodies only when they are activated by T cells. In cell mediated immunity, the effector T cell, it activates the macrophage that has engulfed antigen, and also this effector T cell can stimulate the effector T cytotoxic cell that bind antigen, and this effector T cytotoxic cell destroys the infected host cells. So, those cells presenting viral proteins or other abnormal proteins that signify danger, they are getting destroyed. The acquired immunity can be considered to have two limbs. The acquired immunity is having active immunity and also passive immunity. What is the difference between active immunity and passive immunity? Active immunity, when the host is exposed to antigens, there will be production of antibodies. So, that is known as active immunity. What about passive immunity? Passive immunity happens by direct entry of antibodies. When ready-made antibodies are entering into an organism, then it will result in passive immunity. So, acquired immunity is having two types, there is active immunity and passive immunity. Active immunity will take long time to develop because there will be activation of the immune system by the entry of antigens, whereas the passive immunity is comparatively quick because here the entering antibodies are involved in maintenance of immunity. So, it is comparatively quick. But we can also say that active immunity is long lasting or it will have a long term effect. But when you consider the passive immunity, it is short lived because when antibodies are used up, then the immune system will decline. So, here you can see how adaptive immunity is classified. You can see it here, there is active immunity and passive immunity, and further the active immunity is classified into natural and artificial. Like that, passive immunity is also classified into natural and artificial. What is natural type of active immunity? Natural type of active immunity is attained when the microbes are entering into our body or whenever there is an infection. So, by way of infections are supposed to be natural. So, by way of infections, when microbes are entering, that will activate our immune system and they are considered to be antigens. So, that is a natural type of active immunity. In artificial type of active immunity, we deliberately inject antigens, but remember these antigens are in a killed form or in an inactivated form. So, killed or attenuated antigens, when they are injected into our body, it will also activate our immune system. So, this is what is called a vaccination. So, vaccination come under artificial type of active immunity. Now, what about passive immunity? It is also divided into two types, natural and artificial. As I told you, passive immunity is by administration of antibodies, and these antibodies come sometimes in a natural manner. In the case of a fetus obtaining immunoglobulin G, which is crossing placenta from mother, that is called a natural type of passive immunity. So, a fetus is getting IgG from mother through placenta, that is natural. And like this, in the case of a newborn, when the newborn is nourished by milk produced from the mother, that is also quite natural. And in the case of milk from the mother, there will be IgA. So, IgA, which is present in the first milk or colostrum, it will give a passive immunity to the newborn. So, a passive immunity is given to fetus by IgG and passive immunity is given to newborn by IgA. Both belong to natural type of passive immunity. And there is another one, artificial. What is artificial type of passive immunity? Suppose there is a snake bite. In the case of snake bite, it is an emergency situation. In that emergency situation, we cannot wait for a long time. We cannot wait for the immune system to get activated. So, at the time of snake bite, we will give preformed antibodies, a preparation of preformed antibodies directly, and that is considered as artificial type of passive immunity. And in the case of tetanus, again preformed antibodies are given. So, that is also artificial type of passive immunity. So, passive immunity can be natural or artificial. Edward Jenner is the person considered to be the inventor of vaccination. The concept of vaccination was introduced by Edward Jenner. He experimented in, his experiment was on the smallpox. He found that in a village, lot of people, they are getting affected by smallpox, but milkmaids are not getting, or they are not affected by smallpox. He began to think why these milkmaids are not getting affected by smallpox. His conclusion was that all these milkmaids, they are in close association with cattle, and they all have cowpox in them. They all have a mild form of cowpox. So, the cowpox antigen is actually giving them immunity against smallpox. He found that a small dose of cowpox antigen will give immunity against the smallpox. Now, we are going to study how the T cells are activated. T cells are activated by the entry of antigen. When antigen enters, or whenever a foreign microbe enters, first it is ingested by macrophage, and this macrophage will process that particular antigen. You can see it here. After ingestion of the macrophage, after ingestion of the antigen, the macrophage will process that antigen, and now this cell is known as the antigen presenting cell, and this antigen presenting cell is presenting the antigen to the T cell. And now what happens? You can see it here. The T cell is activated and it is producing its effector cells. It will produce a clone of cells, and each type of T cell will, each effector cell will have its own function. There is a category called killer T cell. Killer T cells, they are directly destroying antigens. There is another category called helper T cells, and helper T cells, they are the T cells, as I told you earlier, which will activate the B cell, and then only the B cell can produce plasma cells, and plasma cells, they are the cells which produce antibodies. Now, here there is another effector T cell called suppressor T cell, and what is the role of suppressor T cell? It will inhibit the T cell and B cell. We can say that it is involved in prevention of autoimmunity or immune responses against our own cells. They are actually avoided or prevented by suppressor T cells. Another category of T cells, they are called memory T cells. And what is the role of memory T cells? Memory T cells will memorize the first encounter of the antigen, and when there is a second encounter of the same antigen, they are capable of producing a secondary immune response. So, here you can see the activation of T cells here. So, now we are going to study the structure of antibody. What is antibody? Antibody is considered as an immunoglobulin. An immunoglobulin, it is having 2 heavy chains and 2 light chains, or we can say it is having H2L2 condition. Heavy chains because there are more number of amino acids, and light chains because there will be lesser number of amino acids. So, a typical structure of antibody molecule is shown here. You can see that heavy chain is having 2 portions. There is a variable region and constant region, and also the light chain is also having 2 portions, the variable region and constant region. Here on the variable region of the antibody molecule, we can see the antigen binding site. Antigen binding site will act as the paratope or it is a region where antigenic determinants are getting attached. What are antigenic determinants? Antigenic determinants are on the surface of antigen, and we can also call them as epitopes. So, these epitopes or antigenic determinants, they actually bind with specific antibodies. So, here you can see the region where the epitope is binding. Epitope will bind with antigen binding site over here. Okay. So, antigen binding site is present on the variable region of the antibody molecule. You can see here there are lot of disulphide linkages. Disulphide linkages, they are helping for maintenance of stability for the antibody molecule. Antibody molecule is stable because of these disulphide linkages, and they are of two types. There are intra-disulphide linkages as well as inter-disulphide linkages. What is intra-disulphide linkage? When the disulphide linkage is present within the same polypeptide, it is intra-disulphide linkage. So, this is an intra-disulphide linkage. What about this one? So, these are inter-disulphide linkages, they link between two different polypeptides. So, the stability of antibody molecule is by way of disulphide linkages. Antibody molecules belong to 5 different classes, and the different classes are IgG, IgM, IgA, IgD and IgE. And all these 5 different classes, they have different sorts of functions. Vaccines belong to the active immunization, and vaccines are of different types. Vaccines, there is first generation vaccine, there is second generation vaccine, and also there is a third generation vaccine. First generation vaccines, they are produced by conventional methods. They are produced from the whole organism by conventional method. And second generation vaccines are recombinant DNA vaccines, or we can say that these vaccines are produced by recombinant DNA technology. And there is a third category called the third generation vaccine, and these third generation vaccines are synthetic vaccines. They are directly synthesized in the chemical laboratory. So, 3 types of vaccines are there, or we can consider 3 generations of vaccines. And of these different vaccines, the second generation vaccine, an example is given here, it is hepatitis B vaccine. In the case of hepatitis B vaccine, how it is produced? Here you can see this is a hepatitis B virus. You can see the antigenic determinants or certain glycoproteins on the surface of this particular virus. Actually, these are the surface antigens which are responsible for activating the immune system. So, how we can produce a vaccine from this virus? Here you can see, first of all, we are isolating the gene coding for surface antigen, and this gene coding for surface antigen, it is inserted into the yeast cell by using a vector. So, yeast cell will act as the host here. So, the modified yeast cells are producing the hepatitis B surface antigen. You can see here yeast cells are undergoing multiplication and they are all producing copy of the surface antigen. So, now from these yeast cells we can isolate the surface antigen and it will work as a vaccine for you. So, further purification and separation will be done, and commercially we can use hepatitis B vaccine. So, hepatitis B vaccine is an example for second generation vaccine. So, here we are not directly introducing the hepatitis B virus, we are not doing that. we only isolate the gene which is responsible for the surface antigen, and by using this recombinant DNA technology, or by depending on the vector and the host cell, we are producing this vaccine. So, that is a method of production of hepatitis B vaccine. Our immune system is also subjected to certain disorders. An example for immune disorder is allergy or hypersensitivity. What is allergy or what is hypersensitivity? Allergy is actually an exaggerated response of the immune system to certain antigens that are present in the environment, and any substance which is capable of causing allergy, we can call it as an allergen. In response to these allergens, our body will produce certain antibodies. I told you there are 5 different classes of antibodies, and of them IgE type or immunoglobulin E type will be produced in the case of allergic response. What are the common examples for allergen? Dust particles, mite, pollens, animal dander. So, any material can form or can act as an allergen. The symptoms of allergic reactions are sneezing, watery eyes, running nose, and difficulty in breathing. What is the reason for the characteristic symptom of allergy? You might have noticed in allergic situation there will be itching. There will be itching on the skin, and it is mainly because and also there will be rashes developed on the skin. All these symptoms of allergy, it is due to the release of histamines and serotonin from a type of cells called mast cells. Mast cells are present in connective tissue. So, they are responsible for this allergic symptom. And how you can reduce these symptoms? All these symptoms of allergy can be reduced by introducing antihistamines. So, against histamine we are introducing antihistamine, adrenaline and steroids. So, by introducing these drugs we can reduce the symptoms of allergy. Here in this picture you can see an allergic response. So, what happens during an allergic response? Here you can see there is an antigen presenting cell over here, and this antigen presenting cell is interacting with antigen, and it is producing some surface proteins, and it is now going to activate the T helper cell, and the T helper cell is producing interleukin 4, and that interleukin 4 is activating the B cell, and the activated B cell will now produce plasma cell, and the plasma cell production will increase the amount of IgE or immunoglobulin E in blood. And what happens to this IgE? It will activate the mast cell. As I told you, mast cells are activated and they will produce various substances. The symptom of allergy is mainly due to histamines, but there are a lot of other substances produced by the activated mast cell, and further there will be symptoms of allergy. So, here in this diagram you can see the immediate phase of reaction, that is after a few minutes. After a few minutes there will be sneezing, nasal discharge, and etc. There will be red color, readiness and itching. In the last phase of the reaction, after several hours, you can see due to the involvement of cytokines, eosinophils they are releasing leukotriene enzymes, and there will be edema, induration and bronchoconstriction. So, this will happen after several hours, and sometimes this may even lead to the injury of the epithelial cell. So, there will be remodeling after a few months. So, there will be injury, there will be considerable injury to response and repair. So, there will be injury and followed by that there will be response and repair. Another disorder of immune system is autoimmunity. This is a condition happening when our immune system is turning against our own cells. You know, our immune system has got a capability. It is able to distinguish between self and non-self. Our immune system will attack only against non-self, only against a substance which is coming from outside. It will never attack our own cells, our own proteins. So, our own cells, there will be a discrimination between self and non-self. But in the case of autoimmunity, due to some genetic and unknown reasons, our own cells are being attacked by our immune system. So, this is what is called autoimmune disease. Examples of autoimmune diseases are rheumatoid arthritis, Graves' disease, multiple sclerosis, etc. So, here in this picture you can see the role of the antibodies are generated due to the presence of antigens. So, the role of antibody is always to respond against non-self. Here you can see in the normal case antibodies are acting against the bacterial cells. So, immune response is generated against the bacterial cell. But here you can see in the case of autoimmunity, in the abnormal situation, antibodies are attaching on the body tissues and the body tissues are also destroyed. So, this is a very serious case of immune system disorder and we can call it as an autoimmunity. Human immune system is consisting of different components. What are the different components of human immune system? The immune system which maintains our immunity is composed of lymphoid organs, tissues, cells and also some soluble molecules called antibodies. So, all these components work together to maintain our immunity. Here you can see lymphoid organs are being classified into two types. What are they? They are classified into primary lymphoid organs and secondary lymphoid organs. You know the difference between these two? Primary lymphoid organs are the lymphoid organs in which lymphocytes are produced and they are getting matured. I told you earlier, B cells are produced from bone marrow and they are matured in bone marrow itself. What about the T cells? T cells are produced from bone marrow, but they will be matured in thymus gland. So, both of them are getting produced, they are getting produced in bone marrow, but the maturation site is different. So, the region from where these cells are produced and they are getting matured, that is called primary lymphoid organ. What about secondary lymphoid organ? Secondary lymphoid organs are the lymphoid organs to which lymphocytes will migrate. Lymphocytes after their maturation, they show a migration. They will migrate to certain other lymphoid organs and these lymphoid organs are called secondary lymphoid organs. And after reaching these secondary lymphoid organs, they are capable of interacting with antigen. So, here the difference is shown. Primary lymphoid organs are bone marrow and thymus, and also after maturation lymphocytes are migrating to certain lymphoid organs and we call them as the secondary lymphoid organs. What are the examples for secondary lymphoid organs? Spleen, lymph nodes, tonsils, Peyer's patches of small intestine and appendix. So, bone marrow and thymus, they are the examples for primary lymphoid organs. Spleen, lymph nodes, tonsils, Peyer's patches and vermiform appendix. They are the examples for secondary lymphoid organs. So, within the secondary lymphoid organs, actually these lymphocytes, they undergo an interaction with antigen, and after interacting with antigen, they are capable of producing effector cells. In this picture, you can see all the different types of lymphoid organs, not only primary but also secondary lymphoid organs are also shown here in this picture. You can see all around this our body, you can see certain structures called lymph nodes. Lymph nodes, they belong to the secondary lymphoid organs. And also you can see MALT. What is MALT? MALT is mucosal associated lymphoid tissue. So, associated with our digestive tract, urinary tract, reproductive tract, etc. So, all these major tracts they are lined by certain type of lymphoid tissues, and these lymphoid tissues are called mucosal associated lymphoid tissue. Now, it is time for us to discuss some questions. First question: from the given clues, identify the concerned body part or tissue. Statement X: it is a large bean shaped organ containing lymphocytes and phagocytes. Statement Y: it constitutes about 50% of lymphoid tissue in the human body. The third statement: lymphoid tissue also located within the lining of major tracts like respiratory, digestive and urogenital. So, you have to identify the concerned body part according to the statements given. Here is a set of options. So, can you find the answer here? Which is that bean shaped organ with a lot of lymphocytes and phagocytes? Yes, that is spleen. Spleen is a lymphoid organ. Spleen is also considered as the graveyard of RBC. So, spleen is a secondary lymphoid organ in our body. What about for second statement for Y? More than 50% of the lymphoid tissue in the human body is composed of. It is composed of MALT. What is MALT? Mucosal associated lymphoid tissue. What about the third statement? The lymphoid tissue located within the lining of major tracts like respiratory, digestive and urogenital tract. That is again MALT. So, here we can select the option B. So, according to statements, according to descriptions, our correct answer will be option B. Coming to the second question, identify the wrong statement regarding innate immunity. So, here 5 statements are given. First one, it is a non-specific type of defense. Yes, innate immunity is non-specific. So, this is a true statement. It is present at the time of birth. This is also true regarding innate immunity. Innate immunity is by genetic makeup of an individual. It is from the birth itself. We are all having innate immunity. What about the third one? It is accomplished by providing different types of barriers. There are 4 barriers. This is also a true statement for innate immunity. There are 4 barriers, you know them: physical, physiological, cellular and cytokine barriers. What about the fourth one? It can recognize and specifically eliminate foreign microbes. Yes, this is not a true statement, this is a wrong statement, because specificity is applicable for acquired immunity or adaptive immunity. So, statement 4 is wrong. What about statement 5? It is available by virtue of genetic makeup of individual. This is applicable for innate immunity. So, our correct answer will be option D. So, from the options we can select option D, because only the 4th statement is wrong here. Coming to question number 3, Identify the correct statements. Again 5 statements are given. We will read each statement. Acquired immunity is not pathogen specific. Is it true? It is wrong. Acquired immunity is adaptive immunity and it is a specific type of immunity. So, this is a wrong statement. So, first one is wrong. What about the second one? Second encounter of a pathogen produces a response called secondary response and it is of low intensity. This is again wrong because the secondary immune response is always of a high intensity. So, this is wrong. What about the third one? Second encounter with the same pathogen elicits a highly intensified secondary or anamnestic response and it is called memory. Yes. So, memory is because of the ability to identify that particular antigen that has already encountered, that has already entered into the immune system. So, this is a true statement. What about the fourth one? T lymphocytes are producing an army of proteins called antibodies. This is wrong because it is B cell producing antibodies. T cells are not producing antibodies, but they can activate the B cell to produce antibodies. What about the last one? B cells themselves do not secrete antibodies, but help the T cells to produce them. This is a wrong statement because actually T cells are not capable of producing antibodies. Actually B cells can produce antibodies. So, the correct way of if you are going to correct this sentence, it will be like this: T cells themselves do not secrete antibodies, but help the B cells to produce them. So, here fifth statement is also a wrong statement. So, from the options you can pick answer. The answer will be option B, because only the third statement is true here. All the other statements 1, 2, 4 and 5, they are all wrong. Coming to fourth question, which one is true regarding antibody? Statements are: antibody molecule has 4 peptide chains, 2 small called light chains and 2 longer ones called heavy chains. This is a true statement. Constant region of antibody is having antigen binding site. It is wrong. Actually, the variable region of antibody is having antigen binding site. What about the third one? 4 intra disulphide linkages are present on the heavy chain with 1 in the variable and 3 in the constant region. This is also true. If you observe that diagram of a typical antibody, you can see the intra disulphide linkages which are present in the same polypeptide. In the case of a heavy chain, this is a true statement. You can see 1 intra disulphide linkage in the variable and 3 intra disulphide linkages in the constant region. So, this is a true statement. What about the fourth one? Allergic responses are evoked by IgE. This is true. Fifth one, IgA present in colostrum will provide a passive immunity to the newborn. This is also true. So, from options you can pick option E, all except 2. So, only the second statement is wrong here, because in the second statement, as I told you, it is a constant region with a antigen binding site. That is a wrong one, because variable region of antibody will have the antigen binding site. Now, a question based on the diagram. In this question you can see the labeled portions. A is a antigen binding site. Antigen binding site is also called paratope. What about B? B is a portion of the light chain, it is a variable region of the light chain, and C is the constant region of the light chain, and D is the heavy chain, and E corresponds to the disulphide linkages. So, our answer will be option C. Now, coming to question number 6, passive immunity of the following is. So, here in this question you have a set of options. So, from these options you have to pick the passive immunity. What is passive immunity? It is the immunity attained by entry of antibodies. Directly when antibodies are entering into our body, we are getting a passive immunity. So, option A, antibodies IgA in colostrum protecting the infant. This is passive because antibodies are entering. What about option B? Fetus receives IgG from the mother through placenta during pregnancy. This is also passive because antibody is coming, and you can say that both A and B, they are natural type of passive immunity. What about option C? In the case of snake bite, we are giving preformed antibodies, that is also passive. So, this passive immunity can be considered as an artificial one because we are doing from our part. Actually, we are artificially introducing antibodies. So, this is artificial type of passive immunity. What about option D? Hepatitis B vaccine produced from yeast given to an organism for protection. This is active immunity because here vaccination is represented. In the case of vaccination, we are introducing killed antigens or attenuated antigens. So, here hepatitis B vaccine is a recombinant DNA vaccine. It is a collection of surface antigens that are prepared from hepatitis B virus. So, we can say that this option D is a type of active immunity. So, our answer will be option E, all except D. Question number 7, select the true statements regarding hypersensitivity. What is hypersensitivity? It is allergic response. So, statements 1, antibodies produced in response with allergens are of IgM. That is wrong, it is IgE involved in allergy. Allergy is due to the release of chemicals like histamine and serotonin from mast cells. That is true. Antihistamine, adrenaline, steroids, they quickly reduce the symptoms of allergy. This is also a true statement because as I told you, we can reduce the symptoms of allergy. We cannot cure allergy, but we can treat allergy. Dilation of smooth muscles and constriction of arterioles of bronchioles are characteristic of asthma. Asthma is also a type of hypersensitivity, or it is also an allergic condition. But here in this statement, smooth muscles they say they are undergoing dilation. It is wrong. Actually, smooth muscles undergo constriction and arterioles they undergo dilation. Arterioles of bronchioles they undergo dilation here in asthma. So, this is a wrong statement. So, we can say that statement 1 and 4 they are wrong. So, the correct answer will be an option with 2 and 3. So, the correct answer will be option D. So, in this session we have discussed the various components of immune system. So, in this session we have discussed about the different characters of immunity and also the various components of human immune system. Thank you.